cutting through the calvarium. The idea is to remove the calvarium, cut through the occipital bone, and connect the foramen magnum. So make the same cutting through or at the same level as we cut the scalp, and then make a wedge incision from the cut end of the calvaria and open into the foramen magnum. The instruments needed: you need a glove, hammer, scissors, scalpel blade fitted on a handle, bone cutter, chisel, sharp plier, dissecting forceps, hand saw. Then we have bone cutting machine. Then your dressing: long sleeve, long lab coat, gloves, and then protective apron. So using the combinations of the, you can cut around and then remove the vault of the cranial cavity, the calvarium. We discussed the external surface, and that is the internal surface. So within the internal surface, the following can be identified. We have the external table. You have the internal table, which is made up of cortical bones. Then we have the inner bone marrow, or the diplo. The diplo contains bone marrow, produce red blood cells. So the extent of the diplo is much more wider in people like sickle cell diseases, where reproductions of red blood cells continue. We have the sulcus for superior sagittal sinus. You have the frontal crest and a number of picts for arachnoid granulation. A number of picts for arachnoid granulation. On the lateral side, we have the groove for middle meningial artery. At the posterior or the occipital side, you see internal occipital protuberance. That will continue down as internal occipital crest. Then the opening of the parietal foramen. So with this calvarium removed, we now expose the covering of the brain. The dura mater. That's the dura. The brain covering, or the meninges, comprises three membranes. The dura mater, or pachymenes, then leptomeninge, which comprises two, the arachnoid mater and the pia mater.